Hi everyone, welcome to my presentation at Research on Demand for APS 2021. I'm Renato Carvalho and today I would like to share my research in the characterization of 3 new species of plant pathogenic bacteria causing diseases in ornamentals in California. Ornamental crops moves an expressive market in the United States. The market includes fresh flowers and nursery products such as flower pots, trees, and bushes. Unfortunately, the ornamental production can be tainted by plant disease such as leaf spots, leaf blight, rusts, galls, and many others caused by plant pathogens such as bacteria and fungi. Although leaf spot has been reported in ornamental crops, the California Department of Food and Agriculture received samples of hydrangea, magnolia, artillaria, and delphinium showing unusual symptoms. Isolations from these lesions resulted in a white and mucoid bacterial colonies, like Pseudomonas colonies, and pathogenicity of these colonies were confirmed by Koch's postulate. To identify the strains, the genes GORB, RPOD, and RPOB were extracted and used to construct a concatenated tree that we can see here. And based on the multilocus sequence analysis, the strain belongs to 3 species. The first species, here in blue, that later we call Pseudomonas magnoliae with only one representative. Here in orange we have the second species that later we call Pseudomonas californiensis with 2 strains. And finally, here in green, Pseudomonas quasi-caspiana, the third species with 3 strains. Phenotypic analyses such as low path were also performed with the strains. Low path is routinely used for characterization of Pseudomonas bacteria. And starting with leaven production test, bacteria that present the enzyme leaven sucrase will break down the sucrose present in the media and form leaven, as you can see in the picture. The second test is called oxidase activity, where the bacteria that present the cytochrome C enzyme will show a rapid development of a purple reaction in the slides. After the oxidase, the third test is called pectolytic activity. In this test, bacteria able to cause rotting potato shows a positive pectolytic activity. The following test is called arginine dehydrolase. In this test, Thorny's tubes are inoculated with bacteria. The presence of the arginine dehydrolase will break down the arginine and release ammonia, changing the medium color to pink in a positive reactions. Finally, low-path tests for tobacco HR. In this test, bacterial suspensions is infiltrating in tobacco leaves. The development of a necrotic area in the inoculation site is recorded as a positive. The low-path results for the 6 strains isolated from the ornamentals can be seen in the table in this slide, and the results are similar for the strains at the same species. Looking for additional phenotypic reactions with the strains isolated for ornamentals, more tests were done using Biolog. Biolog is a plate with 94 different tests, where 71 of these tests are based on carbon sources and 23 are chemical sensitivity tests. The plates are inoculated with bacterial suspensions and incubated to start the reactions. The fingerprints of the strains are then compared with a vast data bank looking for best matches for the identification. Biolog.com identified the samples as Pseudomonas. However, showed a low similarity at species level with any valid published species. The following test to characterize the strains is called the fatty acid methyl ester profile. Fatty acids are essential components in the cell membranes of the bacteria, and they actually determine the difference structure of lipids and lipid A. Fatty acid profiles are a good target for bacterial differentiation strategies by comparing their profiles. The profiles of the 6 strains isolated from ornamental crops is present in here, and comparisons with data bank show that these strains have close relations with Pseudomonas cichoriae, Pseudomonas viridiflava, and Pseudomonas serine pathovar tomato. Apart of all the phenotypic tests for the characterization for the 6 strains, genomic relationships were also analyzed. And when we talk about the genomic relationship, DNA-DNA hybridization we're using the past to calculate the genomic relationship between two bacterial genomes. And even though DNA gene hybridization is still considered the gold standard for bacterial taxonomy, problems such as lack of database, labels process, and experimental problems to reproduction, this technique is not routinely used anymore. With advances in DNA sequence technologies, sequence of 16S and housekeep genes can be used for bacterial taxonomy, as we could see today in the beginning of this presentation. And moreover, whole genome sequence is now routinely used for the bacterial classification, provide a comprehensive comparison among bacterial strains. Using whole genome sequence, we are able to calculate, for example, the average nucleotide identity between two genomes, and the literature shows that genomes that share less than 95% average nucleotide identity is considered a different species. For this work, DNA were extracted for the whole genome sequencing in the 6 strains. The average nucleotide identity were calculated, and then first, the pairwise ANI among the 6 strains supported the result that the 6 strains actually belong to 3 different species: Pseudomonas magnoliae, Pseudomonas californiensis, and Pseudomonas quasi-caspiana. Moreover, the strains do not share more than 95% average nucleotide identity with valid published representative of Pseudomonas genus, confirming then the presence of 3 new species. Transmission electron microscopy were used to confirm the cellular morphology. First, we have Pseudomonas magnoliae here, where we can see the cells are rod-shaped with multiple flagella. We have Pseudomonas californiensis, also these cells are rod-shaped with multiple flagella. And finally, Pseudomonas quasi-caspiana, where again the cells are rod-shaped with the multiple flagella. A little about the names. Pseudomonas magnoliae, the names are chosen considering Magnolia, the crop of the bacteria were first isolated. Pseudomonas californiensis consider California the geographical site of the first inoculation. And finally, Pseudomonas quasi-caspiana. The name was done considering the close relation of Pseudomonas caspiana. As conclusion, all the performance analysis starting from the pathogenicity tests, the MLSA using GYRB, RPOD, and RPOB, the phenotypic profile by low-path, the outcomes from biolog, the fatty acids profile, the pairwise ANI, the ANI with the representative Pseudomonas genus, the cellular morphology by the TEM support the conclusion of the characterization of 3 new species of plant pathogenic bacteria into the Pseudomonas group. With this, I would like to thank you APS, I would like to thank you Plant Pathology Department in the University of Florida and the California Department of Food and Agriculture. Thank you very much, thank you for watching my video, see you next time.